Chronic hepatitis B infection is caused by the hepatitis B virus, HBV, a member of the Hepatnoviridae family that infects and replicates in hepatocytes. A complete HBV virion has several components required to establish successful infection. At its center is a partially double-stranded DNA genome in relaxed circular conformation, rcDNA, associated with the viral polymerase. This is enclosed within an icosahedral capsid made up of core proteins. The entire capsid is surrounded by an outer lipid envelope in which 3 HBV surface proteins, small, medium, and large, are anchored as transmembrane proteins. These envelope proteins are detected as hepatitis B surface antigen, HBsAg, in blood tests. Importantly, complete HBV virions, also known as Dane particles, comprise only one of the many varieties of viral particles found in sera of patients with chronic hepatitis B infection. Non-infectious viral particles lacking HBV DNA occur 100-fold more frequently than complete virions, while viral particles containing linear double-stranded DNA, or RNA, are less common. Most abundant of all, at up to 100,000-fold excess over complete virions, are subviral particles, lipid membrane spheres and filaments bearing only surface antigens. This vast excess of subviral particles provides a screen of decoy targets to absorb anti-HBV neutralizing antibodies. Overwhelming quantities of hepatitis B surface antigen also contribute to dysfunctional, exhausted T-cell responses in patients with chronic hepatitis B infection, allowing infection to persist. The HBV life cycle begins when the virion interacts with hepatocyte membrane receptors. Binding of the hepatitis B surface antigen to NTCP, a receptor that normally brings bile acids into liver cells, enables cellular entry of the virus. The nucleocapsid is internalized into the cytosol and ultimately imported into the nucleus. Here, the viral rcDNA genome utilizes host proteins to establish itself as a stable mini-chromosome of covalently closed circular DNA, or cccDNA. As a library of all the information needed for HBV replication, cccDNA forms the molecular basis for establishing and maintaining viral infection. The transcription of cccDNA from different promoters generates 5 distinct RNA types. The first, pregenomic, pgRNA, is a template for reverse transcription of new viral genomes, as well as translation of core and polymerase proteins. The second encodes precore protein, which is ultimately processed and secreted as HBe antigen. The other 3 encode the large hepatitis B surface antigen, the small and medium hepatitis B surface antigens together, and the multifunctional HBx protein, respectively. In the cytosol, each pgRNA binds to a newly synthesized viral polymerase. 240 subunits of core protein assemble around this ribonucleoprotein complex. The polymerase reverse transcribes pgRNA, ultimately producing a double-stranded rcDNA genome encapsulated in a mature capsid. Meanwhile, surface proteins are synthesized at the endoplasmic reticulum. Migrating to other parts of the cell, they ultimately converge with mature nucleocapsids for envelopment via multivesicular bodies, MVBs. Enveloped nucleocapsids may be released as infectious HBV virions, which are also called Dane particles. In addition, infected cells secrete numerous other viral products that arise through alternate mechanisms at different junctures of the replication cycle. Alternatively, mature capsids containing rcDNA may be recycled to the cell nucleus to replenish its pool of cccDNA. Moreover, linear double-stranded HBV DNA, produced by aberrant replication in 10% of cases, may enter the nucleus of infected hepatocytes and integrate into host chromosomes. Integrated HBV genomes cannot support full viral replication, but do encode hepatitis B surface antigen, augmenting the cellular secretion of subviral particles. In later stages of the disease, integrated HBV DNA is suggested to account for more than 90% of the transcripts encoding hepatitis B surface antigens, rather than cccDNA. Some direct-acting antiviral approaches to treating chronic hepatitis B infection specifically target certain protein-mediated steps of viral replication, interfering with the production of complete virions. Scientists are currently also investigating approaches that use small interfering RNA, siRNA, complementary to the viral RNAs to achieve their degradation. An siRNA trigger consists of two RNA strands that can be conjugated to N-acetylgalactosamine, GalNAc, which ensures selective delivery of triggers to hepatocytes via the asialoglycoprotein receptor, ASGPR. siRNAs can be administered subcutaneously. In the cytosol, triggers are incorporated into RNA-induced silencing complex, RISC, machinery. In this process, the passenger strand is removed, leaving the guide strand available to recognize target mRNAs. Upon recognition and binding, the target HBV RNAs derived from cccDNA and integrated HBV DNA are cleaved by RISC, triggering their degradation. This approach inhibits the synthesis not only of HBV virions, but of all viral products, including the overabundant hepatitis B surface antigen from both cccDNA and integrated DNA. This decreases their immunomodulatory effects and may contribute to functional cure.